Good afternoon, everyone. Good afternoon. My name is Ed Schlesinger, and I'm the Benjamin T. Rome Dean of the Whiting School of Engineering. And it is truly a pleasure to welcome you all here to the Don P. Giddens Inaugural Professorial Lecture honoring Jeff Gray, Professor of Chemical and Biomolecular Engineering. The Whiting School is home to some remarkably accomplished faculty members, and this lecture gives us an opportunity to celebrate their achievements. It allows us to share with our entire community how highly we value our faculty and the work they do as teachers, as mentors, and as researchers. And it is also a fitting way to honor the memory, or I shouldn't say the memory, but to honor one of my predecessors, Don Giddens, the fifth dean of engineering here at Johns Hopkins, whose career has been defined by his commitment to research and education. Today, Professor Jeff Gray will speak to us about how proteins dance with their partners. A fanciful title, but a pretty fascinating area of research. Jeff's work has huge implications regarding our ability to understand diseases such as cancer, Parkinson's, and Alzheimer's, and also to our ability to develop effective treatments. And while his research is remarkable and important, and I know he will deliver a fascinating lecture, I also wanted to say that Jeff is truly a remarkable member of our Whiting School community. His contributions to the Whiting School and to Johns Hopkins, some of which I'm sure many of you know nothing about, is really valuable, enormously important in terms of advancing the quality, the excellence, the diversity of the Whiting School community and Johns Hopkins community. And for that, too, I want to recognize Jeff and thank him for that as well. With that, I'd like to introduce Kostas Konstantopoulos, Chair of Chemical and Biomolecular Engineering.
Hello, everyone, and welcome to the inaugural professorial lecture of my esteemed colleague, Dr. Jeff Gray. Today is a great day for our Chemical and Biomolecular Engineering department because we are here today with friends and family to celebrate the accomplishments of Dr. Jeff Gray. As ancient Greeks used to say, we as individuals are in debt in our parents for raising us, but we are also in debt in our teachers and mentors for training us to be successful in our professional life. So having said that, I would like to introduce Jeff Gray's mentor, Dr. Roger Bonnecaze, who is the William and Betty Nolan Professor of Chemical Engineering at University of Texas at Austin. Dr. Bonnecaze served as department chair for a number of years till 2013, and he's an expert in the area of complex fluids and nanomanufacturing. And actually, he's the director of nanomanufacturing, which is an NSF-funded center at UT at Austin. So without further ado, let's welcome Roger here.
Well, thank you. And I can't begin to say what an honor it is to have been invited to introduce Jeff, my former student, my colleague, and my really close friend for this Don P. Giddens Inaugural Professorial Lecture. So Jeff graduated from the University of Michigan in 1994, summa cum laude, and I still remember him coming to our recruiting weekend that spring, and I thought after I met him, boy, I would be lucky to have this guy join my group. And I thought it was a long shot because Jeff really wanted to do biological type research, and I was more of a colloids simulation person. But when he came, I don't know exactly what I said to him, but I somehow convinced him to join my group, and it's been one of the luckiest and most fortunate moments of my professional career. He did an absolutely spectacular PhD dissertation and really made major progress in understanding the rheology of strongly interacting colloidal suspensions, colloids in confined spaces, and also self-assembly of colloids onto surfaces. And really a lot of his work is the beginning of a lot of the activities that are done on directed self-assembly of surfaces that goes on today. He then, after he graduated, went on to the University of Washington to work in David Baker's laboratory, where David Baker is really the leader in developing software for predicting protein structure. So the biology call was still there, and I think what Jeff did that is really remarkable is that he brought his very strong understanding of colloidal science and molecular interactions, and I think significantly advanced the fundamentals of the so-called Rosetta software package in terms of those aspects, and really made it an even more powerful tool for making predictions of protein structure based on first principles. And now, after several years at Hopkins and all his successes, I think it's without a doubt he is one of the world's leaders on developing codes and tools for understanding and designing proteins. He's one of several folks working on the Rosetta Protein Structure Prediction and Design Suite, and he's particularly clearly the leader on protein docking, proteins on surfaces, and antibody interactions. So Jeff has won numerous awards in recognition of this: his NSF Career Award, a Beckman Young Investigator Award, the Hodgson Scholar at Hopkins, and he's also a fabulous teacher. He is the winner of the Hopkins Alumni Association Excellence in Teaching, and he has really distinguished himself so much that my department is actually celebrating its 100th centennial this year, and Jeff is one of the 11 centennial speakers that's been invited to speak at our department about his research. You know, as a faculty member, and I'm sure many of the faculty members in the audience know this, the best students take the research places you didn't even imagine. And Jeff did that not once, not twice, but really countless times with me working with him. And every meeting was a pleasure, and I was constantly impressed and excited about the things that we would talk about. He also, from the beginning, even as a graduate student, showed a commitment to teaching. He wanted to supervise undergraduates in my research lab, and he did a great job with several of them who've gone on to PhDs at other universities and startup companies now and other activities. He also, from the beginning, was committed to this idea of broadening participation in engineering and in sciences in general, and was active in that even as a graduate student. And I think one of the things that really distinguishes Jeff when I think about all the students I had is that he was truly as much my teacher as he was my student. I learned so many things from him about a myriad of topics. We would have discussions about politics, social policy, in addition to science, that were truly enlightening for me. And honestly, many of the things that we talked about, I still think about later on in some decisions that I have to make. So with that, it's my great pleasure to introduce, finally, the speaker for the program, Dr. Jeffrey Gray.
Wow, there's a lot of people here. Thank you, Ed and Kostas, for making it possible to celebrate this moment, and thank you, Roger, for coming down and saying all those kind things. I've got to say back to Roger. Roger, you were really the model for me of what a professor is like, because working with you is when I learned what a professor does, and I think there are probably three big themes from that. One was going after the most interesting and impactful problems that you can, and using your physical intuition and your theory and your math and your computer tools to solve them as best you can. And second, you set an example for me in service and leadership and responsibility in the roles you took in the department and the lab and the university and in the profession. And that's really been an inspiration to me to see how I can work in areas outside of my lab here. And third, you taught me how to treat people, that we're not just making knowledge, but we're making scholars, I was lucky to have you, and you always treated me like an equal from the first day. And I appreciated that empowerment and that you really nurtured my development as a scholar, as I know you do all your students. And so I've really tried to model what I've done here at Hopkins after many things that I've learned in your lab. So, and I thought I'd say that here because, I don't know, this is a professorship, so that's some of what the professorship's about. I want to talk about, I gave you this title about how proteins dance with their partners, and those of you that know me know that I'm very much into dance. I love it as an art form, whether it's an individual dancing or a couple dancing or a group dancing. It's a phenomenon of life and human existence that is on the meter scale that we can observe and see, and you see lots of connections between people, and it's driven by the energetics, the emotional energy, and the physical kinetic energy between people. And I think there's a huge parallel to that. Life has a lot of complex length scales. As you go down through the length scales, you have different ranges of organization. And at the very bottom, at the angstrom scale, at the scale of molecules, you again have this dance of individuals that come together, that sometimes on their own dance, sometimes dance with partners, and sometimes dance in groups. And it's beautiful, just like real life dance. And so what I want to convey to you today is some of the, actually, let me turn these lights down too. What I want to talk about today is a different talk than what I usually give. Usually I talk about one or two stories and get into the nitty-gritty detail. I want to talk about some of the big themes over the past 13 years since I came to Hopkins of the things we've been working on. And so if I start, but I love the method, so if I start getting into the nitty-gritty details, just give me that face like, you need me to get back up to the high level. So the story starts here with the human genome. So in 2001, I was a postdoc at the University of Washington, and the first draft, the first complete draft of the human genome was published. This was the first time we had the whole list of all the instructions for a human. So we knew there were 23 pairs of chromosomes, but we found out there were 3 billion base pairs. So this would be the equivalent of 3 CDs of information back when we used CDs. And what we didn't know is how many genes we'd found. So it turns out they found about 30,000 genes at the time. Since then, they've narrowed that down to about 20 to 25,000 genes. And what this really is a parts list of all of the parts that make up a human being. So this would be equivalent to, say, if you had a Prius and you had a whole bunch of parts, but actually not even this, you'd really just have a description of the parts and not even their three-dimensional shape. And so you might, from that, want to say, well, what are the three-dimensional shapes and how do all of these parts fit together? Which ones group with which others and how does this go together? And that's where I started getting into looking at how proteins dance and connect. So this is another figure that came around the time I was a postdoc in 2000. In 2000, so let's say you have this parts list. The next thing you know is which parts connect with which others. And there are experiments to do this, to screen the proteins and find the partners. And in 2000, there was a study by a lab, actually, where one of my colleagues, Joel Bader, here at Hopkins, did some work. This is 600 proteins. Each dot is a protein, and each line between two dots is a putative interaction. Turns out a lot of these are wrong because the methods are high throughput and have a high error rate. But at this time, 600 proteins is 10% of the yeast genome. So they had 10% of the genome. They had some of the connections, and a lot of them were wrong. This is if you zoom in, you can see all those connections. Fast forward to this month, a paper just came out in Nature, where a group, including another one of my colleagues, Ben Larson down at the immunology department, a new hire we just brought into Hopkins, looked not just at one organism, but 9 organisms from fungus and fly up through human, and looked at these interactomes in all of those organisms at the same time. If you look at what's common between all of those interactomes and look for the proteins that match each other in the different species, you can come up with about 1,000 complexes. About 2,000 complexes. 1,000 complexes made up of 2,000 proteins that, because they're in all of these organisms, are likely to be complexes that were formed back in ancient cells and that have been conserved through all of these branches of life since then. Some of these are complexes that we knew about, other these are complexes that we don't know. If When you look at the ones that match human and at least one other species, they can narrow down other high confidence interactions that maybe aren't in all but are in several species. And they were able to come up with 16,000 complexes involving 13,000 proteins. So this is half of the human genome. So now with half of the human genome, we can make connections, high confidence connections to other proteins. And so you can imagine the next stage of the work where you actually look at how these proteins come together. There's a huge demand for that, and that's where we started doing our work. So if you wanted to do this experimentally, you need to use some techniques like X-ray crystallography, where you crystallize a protein and shoot X-rays through it, or an NMR machine where you do spectroscopy. And there's a lot of details, but at the end of the day, you can come up with these beautiful structures of proteins, and that gives you a lot of insight into how the proteins work. This is a little bit difficult to do with 20,000 proteins. It's a little difficult to do with 20,000 interactions, and they don't all work. So we look to computation as a way to rapidly go through many, many protein interactions and try to elucidate how these dance together. So the traditional tools is to use molecular dynamics. In molecular dynamics, you put a bunch of atoms in a box. You follow Newton's equation. Acceleration is the force over the mass, and you watch the particles accelerate and move them around femtosecond at a time. It takes a really long time. So this study here is by my colleague in material science, Martin Olmschneider, and he did this calculation of one small protein in a lipid bilayer. It took him a month or two using a whole GPU, which is many, many compute units in parallel on a GPU. So it takes a lot of time to do this traditional follow step by step. It'd actually be pretty nice. That would be watching the dance in real time of all the motions of the protein. But if we're interested in putting these parts together, I really just want to know, what do the parts look like when they finally bind? And so that's where our work fits in. And I'm part of a field known as protein structure prediction and design. So instead of following every instant by instant, we're just looking for the final structure. And that allows us to take certain shortcuts and certain engineering steps to make this more realizable. So this is where I enter the story. This is Rosetta Doc, which is the program I wrote during my postdoctoral fellowship. And you're seeing two proteins bind to each other. And we have a whole bunch of tricks that I don't need to get into, but briefly, a few of them are that we have a multi-scale model. We have a low-resolution search, which you saw where the protein was moving very fast and has single points for side chains. And then there's a high-resolution step that puts all the side chains on. So you can go really fast, and then you can slow down and get the details right. We have pre-computed pieces of protein structure that we, again, don't have to wait a long time for the protein to flip from this spot to this spot, but we can quickly go through that library. We use a cluster of computers that speeds things up, and we also just restart the calculation many times. So here I'm searching really well on the top right. I'll start a completely different simulation on the bottom left and in all kinds of different places and fire up a cluster until we can search all over these protein surfaces to find the very best location where these two bind together. All right, so there's two fundamental problems underlying docking and really under the whole field. So the first problem is sampling. So if we use the dance metaphor, this would be wanting to know where the dancer is moving, what are all the possible poses that dancer can take, right? Either as one dancer or a pair or a group of dancers. And so you need to be able to enumerate all those. So if you know about dancing, maybe you could pull out a couple moves from the Lindy Hop or the tango. If you don't know anything, and you can just follow them a femtosecond along and maybe wait a long time. So that's the first challenge. Can we sample conformations of the protein quickly? The second challenge is known as the scoring challenge, and this is the energetics. Just like in the dancing, there would be certain places where people come together and just fit. We're looking for those fits. So what we want to know is the energy in nature when these two proteins interact. So we need to calculate the energy. Well, nature has an energy function, but we don't really know it. So we have models based on van der Waals and electrostatics and hydrogen bonds to estimate these energies. And that's the second major problem, is to get that energy function as accurate as possible. So during the last 13 years here at Hopkins, we've contributed both of these challenges. The first one, I'm going to show an example of how we attack the sampling problem. So in protein docking, turns out that proteins wiggle when they bind, so it's not just two proteins coming together, but the two proteins are doing their own dance and they're moving. And even a very small motion, here I'm showing an experimental structure of a protein when it's bound and a protein when it's on its own. And you can see there's a small difference here. And when you superpose that onto the complex, what you want to see is when the two proteins come together, you think about the energy of the proteins and the conformational space, and when you get to the right location, the energy should be at a minimum. So that's the two dancers coming together and go, that fits right there. That's the spot. Now, if you have one dancer that's out of shape, it doesn't fit, and that goes away in the energy function. And so very small conformational changes in the protein can confound the algorithms. And this can be a real trick. It's especially a trick if you didn't know the conformation of the protein in the first place and you had to build that from a model. So I'll come back to that in a little bit. So a student in my lab, Sid Chowdhury, attacked this problem. And what Sid did is he pre-generated a whole set of conformations of the protein. So again, this is the protein dancing on its own, and he generated lots of conformations. And then he used that library of conformations during the docking search, switching back and forth between docking moves where he's moving the protein around and moves where he's superposing all of these different backbones of the protein onto its partner. This turned out to be a great speed up, and it also helped us narrow in on the energetic problems. And we ended up being able to dock proteins that were more flexible that you couldn't dock before, and even proteins that were determined with NMR methods that maybe didn't have as much high resolution. By making that protein dance a little bit, we could tease out correct solutions, even in cases where you couldn't before you did this. So the second problem, scoring, or the energy problem, is exemplified by this work by Krishna Kalambi, who looked at the effect of pH. So this is how we typically look at these docking results. This is energy on the y-axis, and this is the distance from correct. So this is a case where I know the answer and we're testing and developing methods. And if the method works right, then you're going to generate structures that are close. So here you're not generating any structures closer than 2 angstroms. And if you look at the lowest energy structures, they're 4 angstroms away. So we're clearly missing the boat here. It turns out that this complex, which is an enzyme called an enzyme inhibitor called Taxi and its enzyme, and it turns out it binds at a low pH, at pH 4.8. And at low pH, amino acids become protonated, and that can change the energetics of the interaction. And so what Krishna did is he figured out the equations for evaluating energies of pH and the chemical potential of putting a proton on. He put a strategy in that would take amino acids like this histidine and put the proton on, and then the energy comes into line, and we get this nice deep funnel with a structure that's closer than 2 angstroms. And so this is the first time actually that anybody had put the accounting for pH into a docking program, which we think is pretty important. So we know it's important in experiment, but it's something that hadn't been captured before in the energy. So we're getting closer to nature's energy function. Okay, so once we've solved some of these fundamental problems, we try to apply this to interesting problems in medicine and biology, and I'm just going to show one example here, again from Sid Chowdhury's work. This is work on HIV. So HIV is the virus that forms AIDS, and it comes in along, it infects the cell, and the cell transcribes the HIV genes and makes this long transcript, and then it has to chop them up. And it has a protein called HIV-1 protease that chops up the proteins. So you can see the enzyme here, and the peptides fit through there, and then it chops up the protein into its pieces. So if you can stop this, it's a way to stop the disease, and many AIDS therapeutics are based on these protease inhibitors. And so what Sid did is he looked at docking peptides into this groove, and he docked not just the natural peptides that HIV has, but also many other peptides and mutants of HIV from a database of the medical researchers that were working on this, and also some of the therapeutic mimetics. And it turns out that if you look at where the natural substrates bind, that's in this red envelope, and the blue is where other mutants can bind. And so it turns out the enzyme is a little bit promiscuous and can do other things. And if you look at where the inhibitors bind, where the drugs are, they're much wider. And so what's happening here is when we have drugs and it fills that hole, if we don't fill it just right, the HIV can mutate away from the drugs, and the peptide sequences also eventually mutate away, and it escapes the drug. And that's why a lot of these HIV drugs don't last as long as we might want them to. So Sid's models helps give molecular insight to how these therapeutics work, how these proteins are interacting, and how you might better design therapeutics to better target this envelope. So a second example of where we apply this and what turned out to be a major thrust in my lab is in antibodies. So you all know antibodies as the smart missile of your immune system. Whenever you have a pathogen, your body produces antibodies to try and match that pathogen. So it turns out you have in you about 1 million different immune memory sequences. These are B cells with antibody sequences, and you have about 1 million different things coded into you, ready to attack some different things. So this is a pretty important molecule to understand. This is a dance that's really important. It's an antagonistic dance because the antibody wants to bind and the virus wants to get away. So yeah, so it's pretty important to understand biologically. It's also important economically because these molecules have such good properties. They've been used as pharmaceuticals. There are almost 50 antibody pharmaceuticals on the market now, and it's an $80 billion market. So if we can develop these better and stronger and more cheaply, it would be an advantage to society. Now, the last two of these to come out are called Praluent and Repatha. They were just approved by the FDA in the last couple months. These are high cholesterol drugs, and statins are one of the largest, most prolific drugs in our country, and this is attacking the same problem, the high blood pressure. And so the neat thing about the antibodies is they bind a particular thing very specifically. So this is binding an enzyme that binds a receptor that binds the bad cholesterol. And so when we understand the biology of the interactions in the cell, we can target exactly what we want to hit. When this biology became known, at least 2 or 3 companies started this mad race to develop this drug because you could tell if we could block this particular enzyme, you could stop you could prevent the cells from not degrading the bad cholesterol, and you could get rid of it quicker. So anyway, so there's great health and economic opportunity in the antibody field. So what did we do? We wanted to know what the structures of the antibodies were, and we wanted to know how these antibodies dock with their antigens. So the first thing we wanted to do is take sequences of antibodies and predict structures. So a lot of the antibodies look the same. We know a lot about them. And I don't want to get too detailed on the algorithm, but we basically put together some canonical pieces of antibodies from known antibodies. And then there are loops at the tip of the antibody that bind the antigen that are a little bit different, and there are libraries for that. And then there's a third loop called the H3 that's really the key loop that has a lot of diversity that allows it to bind a lot of different things, and you have to model that. And so we use all of our tricks, multi-scale modeling and energy functions and libraries of structures to make low energy models of these antibodies. At the end of the day, we can make models that, this is the H3 loop. This is a loop that's about an angstrom away, an angstrom 0.7 away, and they have a decent amount of resemblance to the antibody structures that we test against. But not exactly, right? So when we go to doc, we're going to have to make up for this a little bit. So how good are these models? So one of the neat things we do in the lab, the greater community we're in has a lot of games where we play internationally, where we try to do predictions. So we do this in docking, where there's 2 proteins that there's no known structure, but somebody has determined it, and before they tell everybody, we try to predict it. And so I didn't show that for docking, but for antibodies, we just did last year a challenge where there were a few Janssen and the Scripps Research Institute had new antibodies that had never been published, 11 of them, and there were 7 teams that tried to predict the structure of these antibodies. Daisuke Kuroda and Brian Weitzner in the lab took these on and made these predictions, and this is a graph showing the 10 of the 11 targets and the distance away from correct. And our lab was able to get 4, for 4 of the targets, we had the best model compared to any of the other groups. And for 2 of those, we're underneath the angstrom threshold. So we've got sub-angstrom models of these antibodies coming from sequence alone. So we're pretty pleased with that, to be able to take a new antibody and be able to get a decently accurate model of it. So then what do you do with that model? Well, I want to know how it's going to bind to that antigen. So this is a simulation we call SnugDoc, where we're trying to take the antibody and model to the antigen. And now we know there's problems with our antibody model. We know it's an approximation of reality. So what we do is, while we dock, we're moving the antibody around, particularly that H3 loop and the H2 loop, so we're hedging our bets against the uncertainty in the molecule and the model that we have. All right, so we can also go the other way. We've done some work with design of antibodies, so you might want to design a better therapeutic. We had some funding from DARPA, who was interested in rapidly developing sensors for emerging pathogens. So for example, if you wanted quickly to come up with a sensor for Ebola, you might want to come up with an antibody, and you want it to bind really tightly to the Ebola virus so you can detect positive or negative, does this patient have Ebola or not? And so Jianqing Zhu in my lab, working with Andy Ellington, who has some expertise in using non-canonical amino acids. So these are amino acids that are different than those found in nature. What we did is we looked for places, first of all, we had to make models of the structure, and then we looked for places at this interface where we could hijack this dance. So we took an amino acid that's binding, and we changed it to something that has a chemical reactivity. In this case, this you can put in a peroxide and cross-link for the antibody to the antigen. So now not only do you bind, but you covalently bind to the antigen. It makes it really easy to detect because they want to put these into strips that you can put into harsh environments and just run quick tests on strips. And so we were able to design these non-canonical amino acids to tighten up these complexes toward these sensors. Okay, so to look currently where the field is going and where the outlook might be in the next 13 years, because I've been here 13 years, there's a couple interesting developments in the field. So a study came out called VIRSCAN. This is a study that involves, again, a Hopkins immunologist. And in this study, they can take the whole set of viruses. So there are 206 known viruses that infect humans. They take 1,000 species of these 206 viruses, and with the miracles of DNA manipulations that you can do today, they can put these on a chip and make DNA of all of these different viruses in small chunks. And then they can take a single drop of blood from a patient, screen that over all of these pieces of viruses, and they can detect in your body which viruses do you have antibodies against. So you could see what is your immunological history, what viruses have you been exposed to. And so you can see big population trends, like here's kids under 10 and people over 10, and you can see there's more virus exposure in people that have been around longer, which makes sense. But you can get a broader picture of what's going on in this immune system. So I find that pretty motivating. There's a flip side of that. This is work from George Georgiou's lab. This is measuring the abome, or the antibody, the omics for the antibody, if you will. And what I spent a sabbatical in George's lab in 2013, and they have this beautiful technology where they can take, again, a blood sample, and now they're going to look at individual B cells. So they capture individual immune cells in a droplet in an emulsion. They do some chemistry, and they can sequence each individual cell, so they can get the sequence of each antibody in each cell in this sample from the patient. And so what's exciting about this is a repertoire of antibody sequences, and then we combine this with the virome, where you have the set of all the viruses that a person has encountered, and you've got the whole system, the whole immunological history of the weapons we use to fight the pathogens and all the pathogens. And so to me, this is I think there's gonna be huge things coming from this immunology field in the future. Experimentally, you can look at some of these structures, but there are tens of thousands of these antibodies in these studies, and they're scaling those up, and there's thousands of species of the viruses. So to get that number, it would be really helpful to screen some of them quickly. So that's, again, where I think the computation comes in, because it's quick and easy. So if we could build models of many, many of these antibodies and screen them against these pathogens, we could start asking questions like, when you give a vaccine, what does the body do? How does those antibodies grow and evolve? You could then look at, say, if you're doing the flu virus, you could look at next year, how did the flu change? And if we could get structures of all of these chapters in the fight between the flu and the immune system, you could learn how those develop, and maybe we could get better at picking the vaccines. Maybe we could predict the strains that will come next year and have flu vaccines that hit more often. On the biology side, when the immune system isn't working well, if you have an autoimmune disease, say Crohn's disease or leukemia or even arthritis, you'd want to know, how is the immune system operating differently? What is it stimulating to or not? And again, we could look at these panels of what are the antibodies binding and what are the antibody sequences. And then we have the ability from the computational side to generate structures and try to get molecular mechanisms behind these big questions. So I think there's a huge amount of work and exciting science ahead of us in the antibody field. Okay, that's to wake you up, just to make sure we're all here. And Roger will recognize this. These are movies that I actually made in my PhD thesis. I'm going to do a little change of direction here now. Thought I turned that down. Let's see. So what you're seeing here is colloids. Kostas mentioned that we studied colloids in Roger's lab, and the colloids are coming down on surfaces and forming interesting patterns. My colleague Mike Bevan does this much better now, a few years later. But what I was doing at the time was trying to arrange nanoparticles to make optical materials or electronic materials, and I was publishing papers. Other people were publishing similar papers, and they were claiming that they were simulating proteins on surfaces. And even though I knew very little about biology at the time, I knew that these red and blue spheres were pretty far from proteins. And I knew, it's part of when I decided to come to Hopkins, I knew I had to go to David Baker's lab and learn more about proteins so I could attack this problem. This is my number one problem in my faculty application that I thought I would go after. It's a part of my lab. And I want to talk about that now. So it turns out this is a pretty hard problem, which is why people were doing spheres on surfaces instead of detailed proteins. And so we took a lot of false starts, and eventually we ended up in a little bit narrower field, and that's the field of biomineralization, where you look at biology interacting with solid surfaces. And so now I can get, from biology, I can learn how biology cracks this problem and how we do it. So here's biomineralization. This is our Chesapeake Bay blue crab. It's made of calcium carbonate, calcium carbonate, and some sugars mixed in, and some calcium phosphate, and a little bit of protein. But this is the result of an interaction of a solid inorganic mineral, and it's guided by proteins that help grow this shell. Okay, and there's lots of length scales involved on the cellular level and the nano level. But at some point, there's proteins interacting with these minerals, and we wanted to learn about that. Biomineralization is the one part of my lab where we actually do some experiments in the lab. And I'll show some of those. And then it turns out you can also do experiments in biomineralization at home, because your body does biomineralization in growing bones. And so you can actually do this experiment and watch bones grow. It's a repeatable experiment, and that's even more fun. So I highly recommend this experiment. It's very rewarding. Really, that's the most I know about experiments, but I will show you some other experiments from the lab. So this is what we do with proteins on surfaces. I want to know how the protein binds to a solid surface and what it's doing. So we came up with methods like the docking methods, where I'm exploring the conformations of a single protein and exploring how it can interact with a surface. There's a lot of additional challenges here. You have to define what the surface looks like, so there's a lot of material science involved. This is a polystyrene surface, so this is similar to say a medical implant. So part of when I went to the postdoc to learn about proteins, I thought we would work on devices that you put into humans, and at the time, biomaterials went into a human and sometimes they get infected. The best you could do is just wall it off and not interact with it. Today, with some of my colleagues, they're getting much better about putting biomolecules in these materials and integrating with the tissue, and that's really the dream. But at the time, I thought maybe we'd just do proteins on artificial surfaces and look at how they do. We ended up spending a lot of time with this protein because it was small and easy to study. This is a protein called staphyrin. It's in your saliva. And the mineral there is hydroxyapatite, which is what your bones and your teeth are made of. And this is an interaction between the staphyrin and the hydroxyapatite. And because these predictions are hard to do, and who knows if we're right or not, we teamed up with an experimentalist, Gary Drobny at the University of Washington, who has a solid-state NMR machine and can do experiments looking at one atom at a time, measuring distances and angles. And that's what you're seeing here, are various distances and angles. And together we came up with a set of experimental measurements and then lots of predictions to come up with a full structure that's consistent with those measurements. And it's really the first experimentally supported structure of a protein on a surface. You can't do the X-ray crystallography or NMR on proteins on a surface because the techniques just don't work that way. You don't have enough material on a plane, and you don't have the regular crystallinity. So this got us into this business. We've since done many other proteins and many other surfaces. And then we also tried to flip the equation, going back toward engineering and nanostructures. Could we go the other way, and could we design a protein that could interact with a particular mineral? That's what we wanted to do. So the dream originally was, could we take a microchip and we design a protein that would sit down on a microchip in the way we want it to sit down and talk to that microchip? And maybe the other side is talking to a cell receptor. Maybe there's electrons passing back and through. This is a great idea. All my proposals got hammered because this is way too hard when we started in 2002. But we're getting closer. We backed off. Eventually, the simple problem we decided to look at is calcium carbonate. Again, that blue crab shell. And most mollusk shells. So it's very easy to do in the lab. And a short peptide, which even I can't do the experiments, but I could help some students who could figure out some of the experiments. So if you take calcium carbonate and you put it in a supersaturated solution and start to nucleate it, you can get these beautiful rhombohedrons. And that's nice, so I wanted to try and change this. We wanted to try and stabilize a different face and make this crystal different. So we did that. This is our biomineral activation protein number 4. We had 6 designs. They didn't all do this, but you can see that this is clearly active in the mineralization process, and it makes these steps on the edge of the crystal. Could just be chance. You add something, and it modifies stuff. But if we take that sequence and we scramble it, take the sequence that we designed and we scramble the sequence, and then you add it, you still modify the surface, but much less. You get something much more like the unmodified calcium carbonate. So this is evidence, and we have further energetic evidence from even more experiments, that this particular sequence is interacting in the way with the mineral that we designed and doing something to the mineralization process. So that's pretty exciting to us because this is a pretty hard thing to do on this nanoscale level to craft these structures. So looking toward the future, that's nice, but it's maybe not that useful of a shape. So we wanted to think about some other methods. This is work of a couple current graduate students. One idea is to make an array of proteins, make them dance as a chorus in union. This will help us get a hold of the translational symmetry of the solid, because the solid repeats at a certain interval. We also want to look at the edges and the so-called kinks where the crystal is growing. So this is going to directly interfere with the kinetics of the process as individual ions are coming down. So our next generation of designs involve these kind of structures. The big questions for the next 10 years, first, fundamentally, can we just control this crystal growth and can we shape it? And then I may be getting kind of esoteric with the details here, but we really want to come back to technologies. Could we do this with metals? Could we do it with semiconductors? Could we do it with catalysts? Could you make nanostructured materials that were intelligent and smart and could communicate? If we could combine this, say, Rebecca Schulman and David Gracias and Sharon Garek to have these intelligent biomaterials where you can bind to things and respond and release molecules at different times. So maybe we could do some dynamics, and maybe we could do some spatial control and control and shape minerals at the small scale in real time. So this is the long-term vision for this area. There's a couple new directions I want to mention because we've branched out lately in the lab. One is glycoproteins. So I mentioned there's 25,000 proteins. So there's also a lot of sugars in your cells, in your proteins, and most of your proteins are modified with sugars. And for the most part, we ignore these when we do simulations, and most of my peers do. But they're pretty important, as Mike Beatonbaugh in this department knows. So sugars are pretty complex. They're different than amino acids. They have rings, and they have side chains, and they can be open, and they can be closed, and the glycosidic bond is flexible. This is all in the territory of what we can do with structure prediction methods. So Jason Labonte in my lab has been making these tools to be able to follow these glycans and these glycoproteins. And then there's lots and lots of applications on this, from metabolic disorders and eating and obesity to diabetes. And aging is known to have sugar-related things. This is one that comes back to the antibody problem, and this is a summer intern that worked on this. This is an antibody. So the antibody, the business end attacks the pathogen. The other end is called the FC. It's the constant fragment, and they're all constant and the same, except they're a little bit different, and they bind to a receptor in the immune system. So once you bind an antibody to something, it has to tell the rest of the immune system what to do with it. And depending on what receptor it binds, it can say, attack this thing and kill it. That's what you want to do if you have a cancer therapeutic. Or you might say, chill out a little bit, this is okay. So in, say, arthritis, that's what you want to do, or Crohn's disease, you might want to chill out the immune system. So that's all controlled with this interaction with this FC receptor. And it turns out this interaction is just flooded with sugars. And the sugars are known to play a big role. If you put a fucose on and off, it completely changes the signal in which downstream receptors they go to. So we want to build models of these structures and understand those energetic differences, and then be able to design perhaps improved interactions so you could tailor the drugs to do what you want to do once they've bound to things in the body. The second big new area we've been going into, so we did proteins, we did sugars, is fats. So each of your cells is surrounded by a cell membrane, and those membranes are made up of lipids, and those lipids form this layer that is the boundary between the outside and inside of your cell. You have proteins there that transport things through, that carry signals through and do enzymatic reactions at that location. So there's a third of your genome is membrane proteins. A half of all drug molecules hit membrane proteins. Nevertheless, this is a very poorly understood part of protein science, and there aren't a lot of methods to understand how these proteins dock. So what we decided is to incorporate this into our Rosetta program and look at how with the energetics and the interactions of proteins in the membrane. So here's a study where we're predicting the energy of mutating a single residue on the surface of a membrane protein. These are data from my colleague Karen Fleming in biophysics, and these are experimental predictions. You can see in our first crude models, we have some semblance of experiment, but there are lots of points that are off. So this is an area that needs a lot of work on the energy front. I should also mention the type of application here as we get into the individual genome age of personalized medicine. if you know your genome, if your doctor can cheaply get your genome and you know you have a particular mutation at a particular spot, this is what you would want to do, is look at the structure of that protein, look at that particular mutation and say, what does it do? Is it changing the insertion into the membrane or the stability of that protein or its interaction with the partners? And so these tools, we hope, will inform that day of individualized medicine. Okay, so that's the energy. Here's the searching in the membrane. This is a lot of times membrane proteins are symmetric structures. This is a symmetric tetramer. And we're searching for the best configuration of these coming together. Here's our energy funnel again, and you can see we're not terribly close, but the lowest energy is the closest to the experimental structure. So we have some things figured out along these lines. So I wanted to mention some things about the community and the tools and the way we do things, because when dancing, it's not what you do, it's the way that you do it, right? So one of the important principles we use is that we use object-oriented coding. So the idea here is that when you write the code, you package it down into nice objects. So these objects will contain data or algorithms or manipulations that do certain things. So it means it's an easy piece of software that you can punch buttons and use. So when we looked at this symmetric protein, we didn't actually write code to do symmetry. Somebody else in our community did, and then we were able to plug that in because there was a symmetry object we could plug in. So we try to write these are the objects for our membrane code, and we try to separate out the different pieces of information so that other people can put those parts together. This is a map of all of these different parts and objects in our code. The code is called Rosetta. It's developed by a lot of different labs, and you can see there's a lot of different objects. And so it's really important for us to stay organized and to package things together, and so we can exchange things and amplify our science together. This is the community that does the Rosetta program. This is the so-called Rosetta Commons, which is a loose consortium of 40 labs and about at least 300 researchers. We share in real time on the web our code as we write every day. We share code back and forth. And it's a wonderful way to do science because we can instantly gain from everybody's developments, such as the symmetry or routines to model loops, et cetera, and other people can gain from ours. So it's really important for me to share our science. This is the so-called PyRosetta code. So the Rosetta is written in C++. It's fairly sophisticated. You have to be an expert. Sergey Liskov in my lab with some other collaborators put together wrappings into Rosetta so that you could write it in Python. Python's a very simple language that people can, maybe it doesn't look too simple, but this code here that's just a few lines is what our undergraduates write in about 3 weeks in our class. So we try to make these tools really accessible so that people can learn to do these calculations and work on their individual proteins. We put together a set of workshops on how to do this. So again, we can teach new people to the field how to work on protein structure prediction and design. And then it's kind of fun. In my class last semester, we recorded this, so it's all on YouTube. So there's accompanying videos to go with these chapters in the workbook to learn how to write the code. And so a long-term dream is that this becomes easier and easier as these tools get better so that people can use this science. So while I'm on the topic of community, I wanted to mention that it's been a real privilege at my last 13 year of Hopkins to interact in so many different ways with the Hopkins community and with the outside community. I was able to be a CHE advisor. I was able to work with the Hopkins ChemB Career Network and connect our undergraduates to our alumni. I helped get the newsletters out to our alumni and communicate what we're doing with them. I mentioned the Rosetta community already. There's also been able to be involved, thank you, with a couple outreach programs, the Ingenuity Project and Sawbase, which goes into Baltimore high schools and middle schools and elementary schools. And then I've also enjoyed opportunities to increase the diversity of our community and bring more people into science from different groups. Again, being a professor, there were a lot of challenges, so maybe some comments to some young professors and students who might want to go out to be a professor. The research is really rewarding and really important, but when I look back at the last 13 years, these things are incredibly meaningful to me to be able to bring the community together and be able to build up our program and our school. So I encourage all of you to pursue things along those lines. So to summarize, in the next 13 years, when I double my time as professor here, what do I think needs to happen? Well, first of all, I think we have to focus on improving our craft, sharpening our saw, if you will. We have to be better at what we do. We predict things right. I showed you some of the time. We don't get it right all of the time. We don't have nature's energy function. We certainly don't have it for sugars yet or lipids. or small molecules or other, say, synthetic chemistries, and we'd like to build that energy function so we can really have a clear, accurate model of polymers and interactions between biomolecules. This is going to be a cycle of designing different experiments and testing them and refining our models, and this is going to go ongoing as we attack more problems and test our models in different ways. Second, we want to take all these tools and apply them. The most obvious places are in medicine and nanotechnology. So in medicine, as I mentioned, taking that individualized medicine or precision medicine, taking someone's genome, knowing exactly where their mutations are, from there trying to understand exactly why their phenotype is different, and then trying to understand exactly where you would put in a therapeutic without side effects and directly do the effect that you want to have to change the biology of the patient. So I think there's great potential for that, and I think molecular modeling will play a big role. Second, in nanotechnology, when we're looking at the proteins, building these solid materials from the bottom up, this is Feynman's dream of assembly from the bottom. I think there's a huge future, and there's lots been written on this, from self-healing materials and optical materials and energy sources, and I hope we can play a role in that in helping structure small materials for new applications. And finally, tools for broad use. I mentioned the undergrads can do this in 3 weeks. I'd love to have high school students and even middle school students as we make these tools easier and more reliable to use so that everyone can be a molecular designer, can take their particular protein and study the problem and design solutions to it without having to be an expert in the software. So I hope that's where we go. So to summarize, the fun is in the dancing. Learning new moves, figuring out how to move the proteins, how to move the body, feeling the energy between the proteins and the excitement of the science. I hope I've conveyed that. Dancing at the molecular level will lead us to understand how we were built and how we dock all the proteins together as we figure out our whole proteome, how we battle pathogens and how we can fight disease, particularly through antibodies, how the antibodies fight the pathogens and how we might use them, and how to build small devices and materials, so this surfaces mineralization thrust. So I have a lot of people to thank. My research lab is probably the most important group. I've interacted with many different students through the years. They've all been fantastic. They've all worked really hard, and they've all had great ideas to contribute to this, and we would not be here with all of those influences. So I'm very thankful to all of the students, especially those that are here, for all of the contributions to this work over the last 13 years. I have to thank Roger again for showing me how to do science and how to think about problems. David Baker then taught me about biomolecules and the specifics of proteins, and that formed the foundation of what the work we've done here. This is my colleagues when I joined, or shortly after I joined the university. We were a pretty scrappy group, but they were so formative in teaching me how to do research and how to run a lab and how to raise money and everything else. I thank you all for your contributions. A few years later, they're even scrappier. And there's even more of us, and it's been really exciting to be part of this department. Hopkins has been such fertile soil for doing research, and the expertise in the life sciences and in nanotechnology and nanoscience has just been incredible for us to learn what to do. I've got to make Mark Ostermeyer dance a little bit. Mark's been my partner in protein engineering, and we've had many collaborations and papers together. I didn't mention any of our work together, but I really appreciate working together with him. I also have to make a few department chairs dance. Mike and Joe and Kostas and Kate Staby and Mike Politis, who aren't with us anymore, but who are department chairs and helped mentor me and give me the facilities and support I needed to get things going. In our work, keeping the computers going is critical, and if a graduate student is doing his or her job right, they're going to break the computers at least once in their career, probably several times, and we need people who can make the music that we can dance on. And so Matt Mulqueen is here playing the keys. He's our current system administrator, and he does a fabulous job keeping the stuff running, and we've had a number of different people contribute to that and make our science possible. The Chemby staff similarly have put everything in place, even this event, and made everything happen, and I really appreciate you all making all of our events and the work here possible, and also the staff in the program in molecular biophysics, where a lot of my students come from, and a lot of the work. Again, we collaborate in the School of Medicine, and I get students from the program in molecular biophysics, so these programs have been fantastic. The environment here at Hopkins and the seminars and the types of discussions that go on have been amazing. Funding is critical, so I'm thankful for NSF and NIH, the Beckman Foundation, DARPA, and the Rosetta Commons for funding our work. So as Kostas mentioned, our parents are really responsible for everything you become. My late father here was an engineer, and my mother, who's here, was a teacher, and so I couldn't help admiring them, following to be an engineering teacher, and so I thank them for all the guidance. And finally, my dear wife, Chris, who not only do I like dance, but if you know me well, I'm a pretty anxious person, and so Chris has helped chill me out, stabilize, and help me work through this. And it can be stressful at times for a professor, so thank you, Chris. Again, without her, I probably wouldn't be here. So that's it. Thank you for your attention.
Before we let Jeff go, are there any questions that anyone might want to ask? Yes. Back. Mike and then.
So, I guess you're getting into membrane proteins, and I guess Kalina knows these are a real bitch to work with. So
That's a technical term, right?
That's why I didn't bring my kids today.
But I think she will agree. So, can you talk a little bit about what you need to do differently? Because I know you went through a lot of that in a hurry. So how does the problem change?
Yeah, so the question is, membrane proteins are notoriously difficult. I think that's what you meant.
That's the other term. That's a non-technical term.
And how do we deal with that? Well, luckily I'm on the computer. They're just as clean as ever, so that's nice. But experimental, the data, we don't have as many. So data like Kalina gathered, where does a peptide insert into the membrane or not? Those are data that we can compare against our simulations and see, do we recapture that or not? So one is we have to be a little more creative about the data. There's lots of data about proteins and solutions, 100,000 structures, lots of thermodynamic measurements. So there's lots of places to benchmark and validate our code. In docking, it's funny, we're doing docking, but to my knowledge, there aren't any structures of individual membrane proteins on their own, and then structures when they're bound in a complex. That's the key gold standard data I need to do a blind docking test, and there aren't many structures of membrane proteins, so we don't really have that. But we're getting more and more membrane protein structures all the time, so I know that's coming, and I know as we're able to model these individuals, we'll need these docking tools. On the fundamental side, the thermodynamics is you've got this layer that's hydrophobic, and so we have to change the solvation. You have a solvation potential that wants to bury hydrophobics. We have to indicate that at Z's coordinates it's hydrophobic, and you want to put hydrophobic amino acids there. You have to retune the hydrogen bonds and the electrostatics, and that's the kind of work we're trying to get in to get that physical model right. In fact, there's so many approximations. Everybody's membrane is 30 angstroms thick. That's not true in biology. In the models, in biology, there's lots of different ones. So even modeling different compositions of membranes is something we're trying to think about. Could we run molecular dynamics, extract parameters, and then put that into these fast docking calculations? So we've got a lot of ideas and thoughts, but there's a lot of work to do on this area. Jason, yeah.
Yeah, so you and more generally Rosetta Commons have built up a tremendous amount of infrastructure and just amazing stuff for modeling molecular dynamics. And I wonder how much of this is specific to life on Earth and how much of it will transfer over to alien biology.
Great question. So if you notice we got a little subtle response to that. Oops.
He's got a slide of this.
No, yeah, I've got a slide of the aliens. If you're really sharp, did anybody notice what's wrong with the helix on Rosetta Commons here? It's the wrong stereochemistry. But this will work if you use D amino acids instead of L amino acids, right? So the Rosetta package, there's actually several labs that are working on non-canonical amino acids, D amino acids, and other chemical backbones. So you have to change a lot of what you do. We rely a lot on the energy function of the protein and the structures of proteins to calibrate. So as soon as you change and get out of that protein sphere to alien amino acids and chemistries, you lose all of that touchstone. So then it comes back to, again, you've got to calibrate the energy function and do it. But in principle, we could. In some ways, the sugars and lipids are as alien to us who have been doing proteins for the past 13 years as these other chemistries. But in principle, these kind of approaches would work. In practice, we have to replace all of this empirical data that we have from all of the experiments on biological systems in order to be good at those. Yeah, good question. Roger.
Yeah, great talk. So I was wondering, of course the proteins are really interacting in a solvent, right? There's water, there's ions and things like that. So how are you capturing their effects in your force models you have, energy models, and also how important do you think it is to add those explicitly to maybe sharpen up some of the predictions?
Yeah, good question. So the question is how important is the water? We don't have the water. We use an implicit model for water. It's critically important, right? The water is the background to all these interactions. We use a Gaussian exclusion model, which is a statistical model that says how often are these waters coming together, at what distances, and if you have a hydrophilic atom, how close are those waters, and if you have a hydrophobic atom, how close are those waters. And so with those statistics, we come up with an implicit function that says as you bring two hydrophobic atoms together, you exclude water, and that wants to bring them together. So the model is pretty good at getting hydrophobics to stick together. Where it doesn't tell you is, say, if you have a protein recognizing a DNA and there's a water molecule bridging a hydrogen bond, so a very specific molecular effect where you have these two hydrogen bonds together. We can't capture that because we don't have an explicit model. There's people in the Rosetta community working on that, about putting in particular waters at interfaces and other places. And these are fighting words in the community. We have a review right now from people from the molecular dynamics field who claim you've got to have all these waters and be accurate. Our response to that is we calibrate against structures and against complexes. And sometimes in molecular dynamics, you can't even do that at the level we do because we can hit so many different targets. We test at a length scale that's very different. So it's a current debate how important that is and whether the microscopic water models are good enough to do the kinds of things we're doing. So I hope that answers.
That. Okay, one last question.
Thanks. I want to ask the converse of Mike's question, except not about membranes, which is what you're going to do with more data. So I guess I've heard that the new generation of cryo-electron microscopy techniques will allow you to see uncrystallized proteins with angstrom, well, 3 angstrom scale resolution. So that would suggest you're going to have lots more data. So how is that going to change the role of your simulation?
Absolutely. The cryo-M is really exciting because you're getting larger complexes and you're getting different conformations as the complexes move. It's fantastic. That's going to give us more information to work with. If they're really good, they could replace us because they could do all of this. But there's so much work to be done. I don't think that's a problem. And even if they did, we can take those and have more data to go back and refine our physical models, which I still think is a good goal. But yeah, that development is really exciting because one of the things with crystallization in MR is you're limited to small proteins, and cryo-EM is largely looking at larger complexes, and that's something that we're good at, and there's not too many pieces of data where we can attack those large complexes. Yeah, good question. I'm so glad everybody could come. It's so good to see so many friends and colleagues here, so thank you for your attention.
As is our tradition, I do want to present Jeff with this souvenir of today's inaugural professorial lecture. Jeff, this is for you.
Yes.
Congratulations. Thank you for that. Okay, go ahead. Sorry, talking. I do want to thank Jeff for a wonderful presentation, and I do invite all of you to join us for a reception out in the lobby. Thank you all very much for coming.